Hey guys, Amit Siddiqui here again. So in the previous video, we saw how the electron transport chain worked through the lens of the dam analogy. We saw how batteries such as NADH and FADH2 were able to power those pumps in the electron transport chain so that they could establish a proton gradient which can then be utilized to generate ATP using ATP synthase. Now, how are those batteries recharged? So how are NADH and FADH2, how are they recharged? There are two processes that are involved, that is glycolysis and the citric acid cycle. In this video, we're going to focus specifically on glycolysis. Glycolysis literally means the breakdown of sugars. In glycolysis, we break down glucose into 2,3 carbon chains of pyruvate. And in the process, we generate ATP, which is the battery we want, but also NADH. As glucose enters the cell through the GLUT transporters, it quickly gets turned into glucose 6-phosphate. And there are two reasons why that happens. The first reason is this. Once you attach a phosphate group to glucose, it can no longer escape the cell and diffuse out of the cell. And the second reason is that if you don't change glucose into glucose 6-phosphate, the concentration of glucose will build up inside the cell. And as a result, normal glucose will enter the cell because the concentration inside the cell matches that of outside the cell. And so by changing it to glucose 6-phosphate, we're depleting the concentration of glucose inside the cell, and so more glucose can enter the cell for glycolysis to occur. And so in the first step of glycolysis, glucose gets turned into glucose 6-phosphate because of that addition of that phosphate. And that phosphate is attached to the 6th carbon, hence the name glucose 6-phosphate. And the enzyme that is used is a hexokinase. Hexo meaning 6, right? Or hexose, a 6th carbon sugar. And kinase literally is a type of enzyme that is always involved in transferring phosphate groups. So whenever you see a kinase, you should expect ATP or ADP. And that's exactly what we see here. We get the phosphate from an ATP, we attach it onto the 6th carbon of the glucose to produce glucose 6-phosphate. So glycolysis is analogous to how talent managers in Hollywood find talent. When they find someone who is very talented, they immediately put him in a contract so that they get stuck with this manager. So essentially what we've done in the first step by adding the phosphate to the glucose using hexokinase, and then they give him some plastic surgery to make him more presentable for Hollywood, which is what we do in the second step of glycolysis. We use a isomerase, which is an enzyme that changes the structure of the molecule without changing the number of carbons and oxygens and phosphates, or the number of elements that are already present in that molecule. So if you look at glucose 6-phosphate and compare it to fructose 6-phosphate, we've essentially changed it from a 6 or a hexagon shaped sugar to a pentagon shaped sugar without changing anything else. We've essentially just given this talent a facelift, some plastic surgery, some liposuction to make it more presentable for Hollywood. Now, once we've given this talent some plastic surgery, we need to invest some more money into this talent, and essentially that's what we do. So once we create fructose 6-phosphate, we invest more money by using phosphofructokinase. Remember again, when you see a kinase, you should expect that there are going to be phosphates transferred and that you should expect ATPs or ADPs, which is exactly what we see over here. We see that fructose 6-phosphate gets turned into fructose 1,6-bisphosphate through phosphofructokinase and through ATP, giving us phosphate to that fructose 6-phosphate. Notice how we have two phosphate groups on fructose 1,6-bisphosphate. One is on the one phosphate group is on the first carbon, the other phosphate group is on the sixth carbon. And because these phosphate groups are on different carbons, we call it bisphosphate, not diphosphate. If these two phosphate groups were on the same carbon, so let's just assume that it was fructose 1,1, it would be fructose 1,1 diphosphate because they are both on the same carbon. But because they are on different, we refer to them as fructose 1,6 bisphosphate. So we break up fructose 1,6 bisphosphate into 3 carbons over here. We see that the enzyme that we use to break up this fructose 1,6 bisphosphate is aldolase. And what aldolase reminds me of is the former UFC champion Jose Aldo, who would throw these crazy leg kicks and break fighters down. And essentially that's what aldolase does. It breaks down fructose 1,6 bisphosphate into 3 carbon molecules, one dihydroxyacetone phosphate and the other glyceraldehyde 3-phosphate. But the only 3 carbon molecule that can proceed and progress through the glycolysis process is actually glyceraldehyde 3-phosphate. And so we need to change dihydroxyacetone phosphate into glyceraldehyde 3-phosphate in order for that to go through the rest of glycolysis. We use some plastic surgery, I should say, to change dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. And that plastic surgeon is triose phosphate isomerase. Triose, I always think of tri, so there are 3 carbons and there's a phosphate in it. That's why triose phosphate. And isomerase is the plastic surgeon that's going to do what needs to get done. And so we change dihydroxyacetone phosphate to glyceraldehyde 3-phosphate through triose phosphate isomerase. And now that glyceraldehyde 3-phosphate can go through the rest of glycolysis to produce energy for us. And so glyceraldehyde 3-phosphate gets turned into 1,3-bisphosphate glycerate through glyceraldehyde 3-phosphate dehydrogenase. Remember, when you see a dehydrogenase, you should always expect NADH or FADH2, which is exactly what we see in this step. When glyceraldehyde 3-phosphate gets turned into 1,3-bisphosphate glycerate, what we see is NAD plus being charged into NADH. What we also see is the addition of a phosphate group or inorganic phosphate represented by Pi. And so this dehydrogenase takes that phosphate group and attaches it to the first carbon of the glyceraldehyde 3-phosphate to produce 1,3-phosphate bisphosphate glycerate. Again, it's bisphosphate, not diphosphate, because those phosphate groups are on different carbons. Once we've produced this 1,3-bisphosphate glycerate, we can begin to harvest the energy from that molecule by taking off those phosphates. So the first phosphate is taken off immediately in the next step by using a phosphoglycerate kinase. Again, kinases means that there is going to be phosphates transferred from one molecule to another molecule. And in this case, the phosphate is taken off the 1,3-bisphosphate glycerate. It's specifically that phosphate that's attached to the first carbon and given to ADP to produce ATP, and consequently also produce 3-phosphate glycerate because we've taken off that one phosphate of that molecule. Now we can't take off the next phosphate immediately. What we need to do is give this molecule some more plastic surgery, and we do that by using a phosphoglycerate mutase. A mutase is essentially the same thing as an isomerase. It just changes the structure of the molecule without adding or removing any of the elements like carbon, nitrogen, oxygen, hydrogen, so on and so forth. And so we see that this mutase literally takes one of the phosphate group that exists on the third carbon and shifts it to the second carbon to create 2-phosphoglycerate instead of 3-phosphoglycerate. And now that we have 2-phosphoglycerate, we're going to dehydrate this 2-phosphoglycerate by using a enolase. And you can see that the OH of that third carbon and the hydrogen of the second carbon is taken out and lost in the form of water, and consequently that causes a double bond between the second carbon and the third carbon. And so we create phosphoenol pyruvate from 2-phosphoglycerate. Now that we have phosphoenol pyruvate, we are at the very last step, and we see yet again another kinase. And in this time it's pyruvate kinase, which strips that last phosphate off the phosphoenol pyruvate and gives it to ADP to create ATP and pyruvate. Let's do some accounting. We invested 2 ATPs in the investment stage of glycolysis, specifically in the step where we turn glucose into glucose 6-phosphate, and in the step where we turn fructose 6-phosphate into fructose 1,6-bisphosphate. However, we harvested 2 NADHs in stage 3 and 4 ATPs in stage 3, which was the harvesting stage. So in total, we have 2 ATPs and 2 NADHs generated from glycolysis. In the next video, we're going to cover the citric acid cycle, and you'll get a chance to see where majority of the NADH and FADH2 batteries are recharged. If you found this video helpful, hit like and subscribe, and don't forget to share it with your friends. Don't forget to also comment below if you have any questions or you would like to make some video requests.